Well, the purpose of gene therapy is to correct the aforementioned changes to the genes that have caused or are causing certain diseases by repairing them at the source. It is a technique designed to help restore proper function of abnormal genes by modifying an individual's genetic information found in their cells as a way of preventing, treating, or curing disease. It is the process of, and can include, delivering new genetic material into cells within the body to help it treat or fight disease, replacing faulty or defective genes with ones that are healthy to counteract the effects of the abnormal genes, or deactivating the problematic genes to prevent further disruption. As previously mentioned, a healthy copy of a gene is delivered to the cells inside of the body to replace one that is damaged, to introduce one that is new, or to inactivate a gene that has been mutated. But what we have yet to discuss is how exactly this biotechnology technique works. If you try to insert a gene directly into a cell, it's most likely no longer going to function. This is why, in gene therapy, a vehicle or a carrier called a vector is used, which is genetically engineered to transport and to deliver the healthy gene into the cell, where it is then able to function and perform its appointed task. You may be thinking, what are vectors? Often, vectors are viruses that have been modified in a way that prevents them from performing their habitual role, like causing infectious diseases. Viruses can be used as vectors because they already naturally have the ability to deliver genetic material within cells. They can introduce the new genes by infecting the cells without causing their usual outcomes. Now, the process of introducing and delivering the vector into specific tissue in the body, in an area crowded with defective cells to be absorbed by those cells, is quite simple. However, it is still a difficult task to achieve properly. It is done simply by means of injection or intravenous infusion. This can also be performed ex vivo, meaning outside of the body, by introducing the vector carrying the therapeutic gene to a patient's collected cell sample, and later returning or transplanting it back into their body through the same previously mentioned means. The genetic material being transferred changes the way a specific protein, or a group of proteins, is fabricated by the cell of the patient. It can reduce the quantity of the proteins responsible for causing disease, increase levels of disease-fighting proteins by elevating the production, and even assemble new and/or modified proteins found within a cell. Billy was shocked to hear that there